In this part, I'll deal with nicotinic acetylcholine receptors at the neuromuscular junction. Remember, in the previous part, we discussed synaptic transmission at the neuromuscular junction. Excitation, contraction, coupling in skeletal muscles takes place here. This is the neuromuscular junction, and this is the skeletal muscle, where the contraction takes place. And here we have the vesicles loaded with acetylcholine as the neurotransmitter. This part has been discussed in depth in part 5 on my playlist. Synaptic transmission at the neuromuscular junction, as discussed in video 22 on the playlist. Here's a quick recap. We have the motor neuron. At the end, we have the synapse, and this is the neuromuscular junction. Okay, here again, we have the vesicles loaded with the neurotransmitter acetylcholine. Release of acetylcholine is followed by binding to these receptors. These are the nicotinic acetylcholine receptors, called so because they can be activated by the agonist nicotine. Now, we here have always so-called spontaneous miniature end plate potentials. Miniature end plate potentials. They always take place there. They are small depolarizations. Once acetylcholine is released, we have the graded potential, which is this one here. You see, the more acetylcholine is released, the greater the graded potential. Here I have some notes for you. Under physiological conditions, an action potential in a presynaptic motor nerve axon produces a depolarizing postsynaptic potential, which is an excitatory postsynaptic potential that peaks at around 40 millivolt, more positive than the resting memory potential. This large signal results from the release of acetylcholine from only about 200 synaptic vesicles, each containing 6,000 to 10,000 molecules of acetylcholine. You see, every single vesicle is loaded with acetylcholine up to 10,000 molecules in a vesicle. So now let's take a look at the properties of acetylcholine receptors. It's a heteropentameric structure. You see here, 2 alpha subunits, 1 beta, 1 gamma, 1 delta. Acetylcholine receptor is gated. So acetylcholine gated receptor, it's an ionotropic receptor. You see here again, the pentameric structure of the receptor. Acetylcholine binding occurs at the alpha subunits. We need 2 acetylcholine molecules to bind to these 2 alpha subunits in order to change the configuration of this receptor. Acetylcholine binding results in opening of this receptor, which is an ionic channel. Now let's see about the ion selectivity of this channel. It's highly selective to the neurotransmitter acetylcholine, but not quite selective to a specific ion, as we see from this Nature publication. Now let's discuss why. We see here the channel. Inside the channel, in the mouth, we have a negative cloud, negative ions. So, if, for instance, chloride ions would like to go through, they would not manage because they will be repelled. Okay? Now let's take a look at the cations. For instance, sodium, potassium, and calcium. Sodium and calcium have electrochemical forces driving them inward. And indeed, this channel is wide enough to allow their passage through. Potassium can also pass through, but the electrochemical forces are driving potassium ions outward. And here I have the notes for you. The diameter of this channel here is about 0.6 nanometer, which is large enough to allow sodium, potassium, and calcium to move easily through the opening. Indeed, patch clamp studies have shown that one of these channels, when opened through binding to acetylcholine, can transmit up to 30,000 sodium ions in a millisecond. Conversely, negative ions such as chloride ions do not pass through because of the strong negative charges in the mouth of the channel that repel negative ions. I hope this was helpful.